Hello, Green 9, and welcome to your video on reaction rates. We're going to discuss what actually changes the rate of a chemical reaction, what factors we can look at that will determine how fast or slow a reaction is taking place. First of all, we should talk about just the word of rate, right? What rate means. And rate is basically the how fast something is changing. It is the speed that something is occurring at. So when I'm talking about a chemical reaction, I'm talking about how fast the reaction takes place. Does it finish in, let's say, 5 seconds, or does it take, years, for example? So an example of a chemical reaction that normally has a very slow rate would be something like rusting. It takes a long time, but something like combustion has a very fast rate. That chemical reaction occurs quite quickly. So what factors determine the rate a reaction takes place? And this is something that we cover here in Science 9, but then we actually cover it again in Chemistry 30 and look at it in way more detail at that level. But this is just kind of the basic stuff that you can look out for that will determine the rate of a chemical reaction. Just to give you another idea of a rate, something like speed is a rate. It's how far something travels per unit of time, right? So for example, if I'm talking about something like 50 kilometers per hour, that is a rate. It's how much distance something goes per 1 hour of time. So we're not going to get into numbers for reaction rate, but that's the idea of a rate, is just how fast something occurs, right? So now we're going to go through a list of 5 factors that will determine how fast a reaction is taking place. The first factor that we're going to take a look at is presence of a catalyst. We're going to get into more detail. So I'm just going to read through this list, and then we're going to go through each of these one by one. Presence of inhibitors is actually kind of misleading because that will slow down a reaction. And just the word inhibit is an important one to understand and know. It basically means that you're slowing it down or kind of trying to stop it from happening. You're inhibiting it. Concentration of reactants, we'll look into that. Temperature of reactants, and then finally surface area of reactants. That's a really big one. Okay, so going through these one by one. Presence of a catalyst. A catalyst is a chemical that helps a reaction occur, but it doesn't itself get used up in the reaction. So it's not a reactant because it doesn't get changed. Its atoms don't get rearranged to make something new. Instead, it helps the chemical reaction to occur. So its presence allows the reaction to happen at a faster rate than if it wasn't there. We actually use the word catalyst maybe in life, right? When we talk about you were a catalyst for change or a catalyst for this, it means that your presence, your actions helped that thing to occur. Now, really important again that you understand it isn't a reactant, but it's something that helped it happen, but it does not get used up. To get fancy, we can actually go through an example of a catalyst. So if I take a look at reaction. This is actually chemistry 30 material, so you're getting pretty fancy here. Something like the hydrogenation, okay? And if you talk about hydrogenated fats or hydrogenated oils, that's the same term. It's adding hydrogen as a reactant to something and it reacts. So if I take a look at the hydrogenation of something like C2H2, okay, so acetylene, is what this is called, or ethine, and then I react it with hydrogen, H2 gas. One catalyst that helps this occur at a faster rate is platinum. Okay, when I use platinum as a catalyst, and I often write it above the arrow to show it's a catalyst, then it actually causes those hydrogens to add to make something called ethene, C2H4 gas, much more quickly than if that platinum was not present. Now, the platinum does not get used up at all in this reaction. It's just there, and it helps a reaction to occur. Often catalysts are actually just found by testing out different chemicals with a reaction just to see if it goes faster as a result of that chemical being present. So a lot of finding catalysts is just simply trial and error. As our computer simulations and understanding of chemistry gets better, now it's kind of a thing of you can figure out what catalyst would be best by using things like computer simulations. But in the past and still, Often catalysts are found by just trying stuff out, adding things together and seeing, hey, does this speed it up? Okay, so experimentally determined. An enzyme is a term for a catalyst that is in a living thing. You have many enzymes in your body. In fact, an experiment you can do at home right now. That's right, true science. Take a soda cracker, put it in your mouth, chew it up, and leave it in there for a long time and just keep on chewing. You actually have a enzyme in your saliva called amylase, which breaks down starches into simpler sugars. So the starch that's in that soda cracker will actually get broken down into a simpler sugar. And then over time, after let's say 5 minutes or so, yeah, I know it's a long time for you to be chewing on a soda cracker, but you go through pain for science. After 5 minutes or so, you'll find that that soda cracker starts to taste sweet. And the reason why is because you're breaking down those starches into simpler sugars, simpler carbohydrates. So that is an enzyme that is in your saliva that helps you break down complex carbohydrates, starches, into simpler sugars that then you taste as being sweet. And there it is. An example, we have enzymes in our saliva to act as a catalyst to break down starches into smaller particles and make digestion happen quicker. And just again, if you're curious, it's amylase that is in your saliva. Okay, now, just as a quick kind of thing, you're actually going to see catalysts in a video that I want you to watch that I'm going to link in the description of this video. Maybe wait for that. I'd like you to wait until we get to concentration and then go and watch the video. I'll remind you when we get there. Presence of inhibitors. Inhibitor is like the opposite of a catalyst. It will slow down a reaction with its presence. So essentially it makes a reactant so it's less accessible to react. So inhibitors may be added to foods or medicines so that they don't go bad as fast. They stop a reaction from occurring, the breakdown, the decomposition, by essentially being there. So they inhibit the reaction from taking place. So preservatives are a great example, right? And then they inhibit basically like molds and other things from breaking it down because that's all just chemical reactions. So they stop that from taking place. Concentration of reactants, this is more of a confusing one to understand. So concentration is something that is kind of a hard concept to explain, but it's how much solute I have, so how much of a chemical I have per larger volume of a solution. So the higher the concentrations, the more reactant I have per volume, the faster the reaction is going to be. The video that you're going to watch in a few moments' time shows hydrogen peroxide. So for example, normal hydrogen peroxide that you get in a store is 3% volume per volume. So what that means is if I had 100 milliliters of the hydrogen peroxide, which is H2O2, 3 milliliters of that would actually be H2O2 and 97 milliliters of that would be water. Now, if I had 10% of hydrogen peroxide, then if I had 100 mils of that solution, then 10 mils of that would be the H2O2 and the other 90 mils would be water and so on. Well, as I go to higher and higher concentrations, the rate of reaction will go up. So the 10% will react much faster than the 3%. In the video you're going to see, I think they even have 30%. I'm pretty sure that's the concentration they use, which means that for 100 mils, there would be, whoa, that's fun, what just happened there. For 100 mils, there would be 30 milliliters of hydrogen peroxide. I'm just going to back that up to make it look pretty again. For 100 mils, there would be 30 milliliters of hydrogen peroxide in there, and 70 mils of that would be water. So that would react even faster than all of the rest of them. So the higher the concentration you have of reactants, the faster the reaction is going to go. So an example, if you want to make volcano, the baking soda and vinegar thing that everybody's done in the past to make your model volcano, if you want to do that and have it be a really exciting volcano, you will want more concentrated vinegar, right? Instead of less concentrated vinegar. So you don't want to water down your vinegar at all. And you could actually try this. You could try using vinegar where you have half and half water and vinegar, and then pure vinegar, and then note the reaction with the same amount of baking soda and compare the two and see how they compare. The one with the stronger vinegar is going to go much faster. It's going to create much more carbon dioxide gas, more bubbling than the one that is more dilute. All right, so at this point, I want you to look at the linked video in the description, and it is one from Science Bob, where he's on this TV show, and he shows you elephant's toothpaste, which is a reaction of hydrogen peroxide, where it breaks down into water and oxygen gas. And he does different concentrations, and he also uses different catalysts. He uses yeast, and then he also uses potassium iodide. Potassium iodide is a much better catalyst than the yeast, as you're going to see. Okay, but the yeast is still a catalyst. It doesn't get used up. It's not part of the reaction, but it helps the reaction occur. And then potassium iodide is much more violent, much faster reaction than otherwise. Also note the difference between the concentrations of 3%, 10%, and 30% for the hydrogen peroxide. This is a demo that I wish I could show you in person. It is fantastic. It's really neat to see. Very exothermic as well. Lets off a lot of steam, a lot of heat. So kind of cool to see, but can't show it to you in person. So stop now before you move on. Watch that Science Bob video and then continue with me here. All right, moving on. Next slide, temperature of reactants. As I heat up the reactants, it's going to go faster. So providing it heat gives the reactants more energy, which allows them to be able to collide and react to make product. So higher temperature, quicker reaction, lower temperature, slower reaction. I feel like this one is pretty intuitive, but even actually if we go to that hydrogen peroxide reaction, the hotter the hydrogen peroxide, the faster it is going to actually react. And you can even think about this with cooking, right? When you're cooking, when you provide more heat, the reaction takes place and it wouldn't have happened otherwise. Cooking an egg is a fantastic example. The higher the heat, the faster the egg is actually going to cook. When you look at hydrogen peroxide, the higher the heat, the more it actually breaks down into water and oxygen gas, which is why if you want really long-term storage of your hydrogen peroxide, you actually keep it in the fridge. And in fact, at school, that's what we do is we keep our 30% hydrogen peroxide in the fridge so that it stays strong for longer and doesn't break down. And also because it's in a container that if it was making oxygen gas in that container at a fast enough rate, we worry about it bursting. So temperature of reactants being another one that is really important. Again, higher temperature means a faster rate of reaction. The last one we're going to talk about is surface area of reactants. This just means that the more I have the reactants in small pieces, the faster the reaction is going to occur. This is a big thing in biology that's discussed all the time. The higher the surface area, the better. And that's because with biology, the more surface I have for reaction to take place, the better for that organism. So for example, in the mitochondria, the powerhouse of the cell, the more surface area of the mitochondria, the more cellular respiration that can take place to make energy for the cell. When I take a look at surface area of reactants, if I have, for example, a solid that I'm adding to the reaction, the more broken up that solid is, the faster the rate of reaction because it's teeny tiny pieces. So there's more surface area for the reaction to take place. So more reactants can meet up because I have more surface. Normally for this, we show you a demo with Alka-Seltzer tablets. I don't know if you know what those are, but they're like these circular tablets that when you put it in water, it makes bubbles. So if I crush up the Alka-Seltzer and put it in water, it goes way faster making the bubbles than if I have Alka-Seltzer just as a big pellet and put it in the water, right? So the more crushed up it is, the more tiny pieces, the more area the surface has, okay? And then as a result, the faster the reaction. This diagram here is meant to show you kind of surface area. If I take a look at these larger chunks and I counted the surface area of all these sides, it wouldn't be as much as if I have these smaller chunks. Think about it. In this cube, if I split it up, all the surface area that is inside of here, right? Once I split that up, will now be accessible for reaction to take place when it wasn't accessible in these larger chunks. So higher surface area, a faster rate of reaction. All right, well, that's it for today. You guys enjoy the rest of your day.